Assalamualaikum warahmatullahi wabarakatuh. So today we'll be covering the chapter of immunity. So it's an easy chapter. It's not very difficult. There are just few basic concepts that we need to grasp upon, and starting with what exactly is meant by the word immunity. So they may ask you this question for 2 marks. So it's important for you to understand that it is your body's defense mechanism which recognizes any foreign particle or any pathogen which gains entry into your body and has certain toxic effects. So it's a defense mechanism of your own body which is initiated or which is triggered upon the entry of any foreign particle. And what exactly is meant by a foreign particle? By this, I mean a pathogen, or it could be any bacteria, it could be any virus which gains entry into your body and has certain toxic effects which are harmful for your body. So immunity is your whole defense system. Then there are two types of immunity. Number one is your external defense system, and number two is your internal defense system. So, what exactly is meant by external defense and internal defense? We'll study it in depth. So, starting off with external defense system, it's very basic that you have hydrochloric acid in your stomach, then you have tears in your eyes. So when any dust particle gains entry into your eyes usually become watery. So tears also come under the umbrella of external defense system. And then there is this mechanism of blood clotting. So all three of them are under the umbrella of external defense system. Then you need to remember that how many types of cells of our immune system are there. So you have phagocytes and lymphocytes. So this is a very important MCQ, that which of the following are the cells of immune system. You need to remember that phagocytes and lymphocytes are the cells of your immune system. Okay. Then your body recognizes a foreign particle. By foreign particle, I mean pathogen, bacteria, or virus. They are all the same. Rather, they are the synonyms of one another. Antigen, which stimulates an immune response. So your body is this much sensitive that whenever any foreign particle gains entry into your body, then the entire immunity system or the entire immune response is stimulated. So your body is this much sensitive. Then your immune system stimulates the production of antibodies, which are glycoprotein, and they are derived by B lymphocytes. So these are three important words that are covered in this tiny paragraph, and that are that immune system ultimately stimulates the production of antibodies. We'll study this in the upcoming slides as well, but you need to understand that your immune system in the last step produces antibodies, and they are glycoprotein in nature, and they are derived by B lymphocytes. So we'll study the categories of lymphocytes, the categories of phagocytes. So at this point, just remember the fact that antibodies are made or they're synthesized by B lymphocytes. All right, so we'll study phagocytes first, and then we'll move to lymphocytes. So phagocytes are also the cells of your immune system, and they're produced throughout life by your bone marrow. This is an important MCQ. Then you have two types of phagocytes. Number one, neutrophils, and number two, macrophages. This is very, very important. So you have your immune system, okay? IS is your immune system. Then you have two cells of immune system. Number one is your phagocytes. Number two are your lymphocytes. Okay, then classify phagocytes into further two more categories, and they are neutrophils and macrophages. Okay, so try to make this chart so that you remember that which cell exactly are we studying. Okay, so there are two types of phagocytes, neutrophils and macrophages. And then neutrophils are 60% of WBC. So this is an important MCQ. they may ask you that how much part of WBC is comprised of neutrophils, so 60%. And they travel throughout the body to patrol the tissue. So they basically act like your traffic police. They are traveling in your entire body. They're continuously patrolling your tissue. So you can remember neutrophils like your uh, traffic police, and they're short-lived. So this is an important difference between neutrophils and macrophages. That neutrophils are short-lived. Number two, they comprise 60% of WBC. Number three, they travel throughout the body. They're continuously in the patrolling mode. Then your macrophages are larger. So the other category of phagocytes are macrophages, which are larger than neutrophils. They're found in organs. Important difference. They're found in organs, while the neutrophils are responsible to patrol your tissues. They travel in blood as monocytes. Another difference, that if they reside in your organs, they are known as macrophages. But if they are traveling in the blood, they are known as monocytes. This is a very important difference. Then they are long-lived. Another difference, they cut the antigens which are then recognized by lymphocytes. So if they ask you the function of macrophages, what you're supposed to write? You're supposed to write that they cut the antigens and then they present those to lymphocytes, which are the other category of immune system cells, right? When pathogen invades the body, the cells under attack release histamine. This is a very important detail. Sometimes they ask you to write the entire working of phagocytes for 4 marks. So this is something very important that you need to mention, that whenever your cells are under the attack, those cells release this chemical known as histamine. And then what exactly is the role of histamine? It is to attract the neutrophils, which are continuously patrolling your tissues, and they destroy pathogen by the process of phagocytosis. And they sometimes ask you to define phagocytosis furthermore. Then you need to write that pathogens, which are your foreign particles, they're clustered together. Then they're covered in antibodies, which are produced by B lymphocytes to stimulate the neutrophils to attack them. So they both work hand in hand. The B lymphocytes and then the neutrophils. So B lymphocytes were responsible to produce antibodies. Those antibodies cover all the pathogens or all the foreign particles, and then the neutrophils, which are continuously patrolling the tissues, they come because they're stimulated. They come and then they attack them. It happens because neutrophils have receptor proteins that recognize the antibody and attaches with them. The neutrophil cell membrane engulfs the pathogen by endocytosis. So endocytosis and phagocytosis, both of them are same. You have already studied endocytosis in your chapter number 4, cell membrane. So the process of endocytosis and phagocytosis is exactly the same, but when we're talking about immune system, we specifically mention phagocytosis. Then your digestive enzymes are released by phagocytic vacuole, and then ultimately your pathogen is destroyed. And later on, neutrophils themselves, they also die because they're short-lived. And once they die, they form the pus at the site of infection. So this is what you need to remember for 4 marks. You need to remember histamine. You need to remember that histamine attracts neutrophils. Then pathogens are covered by antibodies. They're produced by B lymphocytes. They ultimately stimulate your neutrophils to attack them. And then it happens because neutrophils have specific receptor proteins. And once your pathogen is covered with antibody, neutrophils recognize them because of the receptor protein. And then the receptor protein of neutrophil, it attaches with the antibody. And then the neutrophil cell membrane engulfs the pathogen. So the pathogen, which was covered by antibody, it gets engulfed by neutrophil, and then the digestive enzymes, they are released by the phagocytic vacuole, and your pathogen is destroyed, and ultimately a pus is formed at the site of infection. Then if you move towards lymphocytes, which are the other categories of your immune system cells, so again, we have two subcategories of lymphocytes as well, B lymphocytes and T lymphocytes. So B lymphocytes are primarily, they are responsible to produce antibodies. Then your B lymphocytes remain in bone marrow until they mature and then they spread in the body. And T lymphocytes leave bone marrow and collect in thymus. So this is a very important MCQ. They do ask that where exactly T lymphocytes are located. So they collect in your, so they are made in bone marrow, then they're collected in thymus where they mature, and the antigens enter the body. A small number of B cells with receptor complementary to the shape of antigen are divided by mitosis. So this is your step number one, that they divide by the process of mitosis. This division is called clonal selection. So sometimes they do ask you that what type of division is this called. So you need to mark clonal selection instead of mitosis. Then clone keeps on dividing and a huge number of B cells are produced in few weeks. Then your B cells become plasma cells which produce antibodies. So this is a very long chain that initially you have lymphocytes, and the other category of lymphocytes is B lymphocytes, which are responsible to produce antibodies, and then they subdivide into plasma cells, and plasma cells ultimately produce antibodies. So other B cells become memory cells and they keep on circulating in the body. So when an infection reoccurs, they divide into plasma cells. So how can you remember this? You can remember this by making this chart. Lymphocytes. Lymphocytes are categorized into B lymphocytes and T lymphocytes. Okay. So B lymphocytes then by clonal selection or by the process of mitosis, they further divide into plasma cells. Okay, so by P and by P, okay, we'll write this completely. Plasma cells. Okay, then these plasma cells they make antibodies. So yeah, so this is the simple wala task which keeps on happening with the clonal selection of B lymphocytes. But then there's something else that happens as well. Your B lymphocytes they divide into plasma cells, and few of the B lymphocytes they divide into memory cells. Okay, so this is an important detail. And whenever you suffer from the same infection again, those memory cells they are already in your circulatory blood. They understand that okay, this is the same antigen which gained entry few months ago, and then those memory cells they divide themselves into plasma cells. So you need to understand that the clonal selection, the process of clonal selection divides B lymphocytes into plasma cells and memory cells. Plasma cells ultimately produce antibodies, while your memory cells, they keep on patrolling your body. And once the antigen regains the entry into your circulatory system, they divide into plasma cells and ultimately into antibodies. Then, for since we studied that lymphocytes are categorized into two, B lymphocyte and T lymphocyte, B and T. Okay, so similarly, T lymphocyte also is categorized into two of the cells, helper T cells, and your killer T cells. Okay, helper and killer T cells. So helper T cells are activated to release hormones like cytokines that stimulate B cells to divide and develop into plasma cells and secrete antibodies. So what is the function of helper T cells? They stimulate, since the name already specifies that they are helping someone. So they are helping B cells, B lymphocytes to divide and develop into plasma cells, ultimately secrete antibodies. Okay, so helper T cells are there to assess the B lymphocytes. But then we have killer T cells as well. And they do not, resonate with the function of helper T cell. They are completely different. They search the cells that are invaded, and when they recognize antigens, they attach themselves to the surface of infected cells and secrete toxic substance like hydrogen peroxidase, killing the body cells. So by the name killer, you need to understand that they kill those cells that are infected. But how do they kill those cells? By releasing these toxic substances like hydrogen peroxidase. You need to remember this word, hydrogen peroxide, that it is released by killer T cells, and this ultimately kills the body cells. Then you need to understand the difference between primary response and secondary response. It's very easy to understand that in primary response, you get the antigen or the pathogen. It gains entry into the body for the very first time, and there are there are no memory cells in those primary responses. For example, in COVID, when the virus of COVID gained entry, the bodies of the infected patients were producing primary response because they never fought with any of the they never fought with the COVID virus before. So it was their primary response. But influenza is something which occurs to people in every other season, in every winter season, right? So they produce a secondary response for influenza. But if there is any new virus or if there's any new bacteria, then the body produces primary response. And in primary response, there are few beta cells. They are specific to the antigen, while in secondary response, it's a faster response because there are already so many memory cells that are present. And those memory cells then divide themselves into plasma cells and then into antibodies. So this is very important. Antibodies, they might give you this diagram and they might ask you to label it. First, you need to understand that this is the structure of antibody and it has two heavy chains and two light chains. So your antibody always has two heavy chains. These are two heavy chains. These are two heavy chains and these are two light chains. Okay. So antibody has two heavy chains and two light chains. Then it has two antigen binding sites. So these are your antigen binding sites and they're always complementary to the shape of the pathogen or the antigen. Okay? So these are two antigen binding sites. Then antigen binding site forms the variable region. So this is your variable region. And then this is your hinge region, which provides flexibility to bind around or to twist around the pathogen. Okay? So hinge region is there to provide flexibility. And what exactly is the function of antibodies? They punch holes in the cell wall of bacteria, causing them to burst. And it makes your antigen less active and easier to engulf. Then they also combine with toxins and neutralize them, making them harmless. This question might come for core maths. It's a very favorite question of your CA, where they give you that write the functions of antibodies for 4 marks. So this is exactly what you need to write. These are 4 different functions of antibodies that they make the antigen less active and easier to engulf. They combine with toxins and neutralize them, making them harmless. And your antibodies bind with virus or bacteria, preventing them to enter the cell. Okay. Also, there's one more MCQ that might come, that what are the bonds of your antibody? So you need to mention that the bonds of antibodies are disulfide bonds. Okay. All right, moving on to active and passive immunity. Active immunity, so there are two categories of immunity, active immunity and passive immunity. So we can make a classification chart as well. Wait, just give me a minute. Immunity. Okay, so there are two categories of immunity, active and passive. Okay, so there are two categories of immunity, active and passive. Okay, then your active immunity again is classified into two more subcategories. Active immunity is furthermore classified into natural active and artificial active. Okay, natural and artificial. So there are two categories of immunity, active and passive. Then they're furthermore categorized into natural and artificial immunity. Okay, so active immunity is gained when an antigen or a pathogen enters your body and then it stimulates or triggers a proper immune response and ultimately it produces antibodies. Okay, so this is something which is very commonly occurring, that you catched any virus or catched any bacteria and then you fell sick and eventually your immune response was activated. And eventually after a few weeks you had ample amount of antibodies. So this is your active immunity. Active immunity is when you actively gain any pathogen inside your body and a proper immune response is stimulated after that. And then there are two categories of active immunity, natural active and artificial active. Natural active immunity is when, this is the normal case that I already stated, that when your antigen enters your body, it produces a proper immune response and you suffer with a great infection. And ultimately, after a few weeks, you see that there are antibodies in your blood. Okay, so this is your natural active immunity. While your artificial active immunity is when an antigen is deliberately injected into your body. And this is usually the case with your vaccinations when there's this proper system of vaccination for infants, right? So what exactly is happening in vaccination? They are being injected with antigens so that their body gets used to that there are certain antigens and they need to produce certain immune responses. They need to produce certain antibodies. And usually after vaccinations, these infants usually they suffer with fever. So that's an artificial means of, injecting an antigen. They did not catch any antigen, rather we injected in them. So this question also comes in your MCQs that which of the following is artificial active immunity? So you always need to mark vaccination. Vaccinations are your artificial active immunity where you are injecting the antigen through artificial means. Then comes your passive immunity. Okay, so passive immunity again is subcategorized into natural and artificial passive. So passive immunity is gained without an immune response. So your immune response is skipped. And ultimately, so this proper immune response, right? We studied how neutrophils patrol your blood, then how macrophages are responsible for the immune response, and we studied the working of B lymphocytes and T lymphocytes, right? So all of that was immune response. But in passive immunity, you directly jump to the last part of antibody production. You directly get antibodies without going through the entire immune response. So hopefully I'm making sense to you that in active immunity, you activate your entire immune response, while in passive immunity, you do not activate your immune response. Your cells do not work. Rather, they directly get antibodies, which is the last step of your immune response. Okay? You directly get antibodies without triggering your immune response. So this is your passive immunity in which your antibodies are directly passed into the body and they're either injected or passed through by breastfeeding, because there are two categories of passive immunity. In natural passive immunity, the antibodies are injected or they are transmitted into the infant through breastfeeding, and in artificial passive they are injected. So antibodies usually they are injected. You must have heard that in COVID-19, the people who were suffering with COVID-19, they were asked to donate their plasma. Why? Because plasma has antibodies, right? So they were directly provided with antibodies rather instead of going through or experiencing the entire COVID virus. They were directly injected with antibodies. So if the antibodies were injected in them, they skipped the immune response full apart, and rather they had antibodies to provide them proper protection against COVID virus, right? Then the vaccination. So vaccination is when antigens are injected. We studied that vaccinations are your artificial active immunity. Then some vaccinations are very effective, but those are less effective. They're administered along with boosters. Vaccination on larger crowd helps to achieve herd immunity. They might ask you what is herd immunity. So herd immunity is when the entire crowd is given certain vaccinations, as in COVID-19, when there were mass vaccination programs. So they were trying to achieve herd immunity. It interrupts the transmission in population. Sometimes people do not respond to vaccines as they might have a defective immune system. Then the virus keeps on mutating. So virus, it's very difficult to produce, vaccinations for viruses because they keep on mutating themselves. And the vaccinations for viruses are not very much effective because they keep on changing their antigen and the memory cells are also unable to identify them. So last but not the least, the last subtopic is monoclonal antibodies. So it's a type of protein that is made in the lab. So monoclonal antibodies are made in lab and they can bind to certain targets in the body, such as antigens on the surface of cancer cells. And there are many kinds of monoclonal antibodies, and each monoclonal antibody is made so that it binds to only one antigen. Okay? They are lab-produced antibodies and they are derived from single clone of B cells. This is very important. You need to remember that monoclonal antibodies, they are produced in lab, and how are they derived? They are derived from single clone of B cells, any B lymphocytes. The hybridoma technique is employed. So you need to remember that which technique is used to make them. So the hybridoma technique is used to make monoclonal antibodies. And what exactly happens is that mice are injected with an antigen to stimulate the production of plasma cells that generate antibodies, the usual case of immune response. Then those plasma cells, they are obtained from the mice and they're fused with immortal tumor cells to form hybridoma cells. Hybridoma because half of the cells are, taken from mice and half of the cells are taken from, they might be the cells of human. So this is why we call them hybridoma. Hybridoma cells are grown in selective medium and screened to identify those producing the desired antibody. These selected cells are then cultured to produce mass clonal antibodies. So they sometimes they give you the question of monoclonal antibodies, and this is exactly what you need to write. All of this can be mentioned in your answer script, and you will get 4 on 4. Okay. So Jazakallah khair for joining, and we are done with the chapter of immunity. Inshallah, we are now left with the last chapter of infectious diseases, and I'll try to upload the recording of infectious diseases in like a week or so. So take care and Jazakallah khair for joining. Allah Hafiz.